Hi everyone, the making of a scientist for grade 10 English.
Hello, Mr. Richard Wright. We wish to know about your life.
Okay. I am an only child, and I grew up north of Reading, Pennsylvania. There wasn't much I could do there. I certainly couldn't play football or baseball with a team of one. But there was one thing I could do there.
What?
Collect things. My mother encouraged me in learning. She took me on trips. And also, she bought me telescopes, microscopes, cameras, mounting materials, and other equipment, and helped him in many other ways.
Now, let us ask her mother what she is saying about Richard Wright. I was his only companion until he started school. After that I would bring home friends for him. But at night we just did things together. Richie was my whole life after his father died when Richie was in 3rd grade. My son and I spent almost every evening at the dining room table. But at night, if he didn't have things to do, I found work for him, not physical work, but learning things. He liked it. He wanted to learn. And learn he did. He earned top grades in school. On everyday things he was just like every other kid. Now let us ask about Richard Wright Butterflies collection. Hi, Richard Wright. Can you tell about your butterflies collection?
Yes. Sure. By the time when I was in the 2nd grade, I had collected all 25 species of butterflies found around my hometown. That probably would have been the end of my butterfly collecting.
Oh. Then,
my mother got me a children's book called The Travels of Monarch X. That book, which told how monarch butterflies migrate to Central America, but that opened the world of science to the eager young collector.
how?
At the end of the book, readers were invited to help study butterfly migrations. They were asked to tag butterflies for research by Dr. Frederick A. Urquhart of the University of Toronto, Canada.
His mother wrote to Dr. Urquhart, and soon Wright was attaching light adhesive tags to the wings of monarchs. Anyone who found a tag butterfly was asked to send the tag to Dr. Urquhart. The butterfly collecting season around Reading lasts 6 weeks in late summer. If you are going to chase them one by one, you won't catch very many. So the next step for Wright was to raise a flock of butterflies. He would catch a female monarch, take her eggs, and raise them in his basement through their life cycle, from egg to caterpillar to pupa to adult butterfly. Then he would tag the butterfly's wings and let them go. For several years his basement was home to thousands of monarchs in different stages of development.
Eventually I began to lose interest in tagging butterflies. It's tedious and there's not much feedback. In all the time I did it, group of dots. Only 2 butterflies I had tagged were recaptured, and they were not more than 75 miles from where I lived.
Then in the 7th grade he got a hint of what real science is. When he entered a county science fair, and lost. It was really a sad feeling to sit there and not get anything while everybody else had won something. His entry was slides of frog tissues, which he showed under a microscope. He realized the winners had tried to do real experiments, not simply make a neat display.
I knew that, for the next year's fair I would have to do a real experiment. The subject I knew most about was the insect.
Insect.
Yes, I'd been doing in the past several years.
So, he wrote to Dr. Urquhart for ideas, and back came a stack of suggestions for experiments. Those kept Wright busy all through e-school, and led to prize projects in county and international science fairs. Then, how did you win?
For my 8th grade project, I tried to find the cause of a viral disease that kills nearly all monarch caterpillars every few years. I thought the disease might be carried by a beetle. I tried raising caterpillars in the presence of beetles. I didn't get any real results.
Not get any result. Then how?
But I went ahead and showed that I had tried the experiment. This time I won.
The next year his science fair project was testing the theory that viceroy butterflies copy monarchs. The theory was that viceroys look like monarchs because monarchs don't taste good to birds. Viceroys, on the other hand, do taste good to birds. So the more they look like monarchs, the less likely they are to become a bird's dinner.
Ebright's project was to see whether, in fact, birds would eat monarchs. He found that a starling would not eat ordinary bird food. It would eat all the monarchs it could get. This project was placed first in the zoology division and third overall in the county science fair.
When did you start your new theory about cell life?
In my second year in high school, I began the research that led to my discovery of an unknown insect hormone. Indirectly, it also led to my new theory on the life of cells. The question I tried to answer was simple.
What was the question?
What is the purpose of the 12 tiny gold spots on the monarch pupa? Everyone assumed the spots were just ornamental, but Dr. Urquhart didn't believe it.
To find the answer, Ebright and another excellent science student first had to build a device that showed that the spots were producing a hormone necessary for the butterfly's full development. This project won Ebright first place in the county fair and in Toronto the International Science and Engineering Fair. There he won third place for zoology. He also got a chance to work during the summer at the entomology laboratory of the Walter Reed Army Institute of Research.
When did you win first place?
As a high school junior, I continued my advanced experiments on the monarch pupa. That year, my project won first place at the International Science Fair and gave me another chance to work in the Army laboratory during the summer.
Oh. Then?
In my senior year, I went a step further. I grew cells from a monarch's wing in a culture and showed that the cells would divide and develop into normal butterfly wing scales only if they were fed the hormone from the gold spots. That project won first place for zoology at the International Fair. I spent the summer after graduation doing further work at the Army Laboratory and at the laboratory of the U.S. Department of Agriculture.
The following summer, after his freshman year at Harvard University, Ebright went back to the laboratory of the Department of Agriculture and did more work on the hormone from the gold spots. Using the laboratory's sophisticated instruments, he was able to identify the hormone's chemical structure. When did you find DNA as the blueprint for life?
A year and a half later, during my junior year, I got the idea for my new theory about cell life. It came while I was looking at X-ray photos of the chemical structure of a hormone. When I saw those photos, I didn't shout, "Eureka!" or even, "I've got it." But I believed that, along with my findings about insect hormones, the photos gave him the answer to one of biology's puzzles, how the cell can read the blueprint of its DNA. DNA is the substance in the nucleus of a cell that controls heredity. It determines the form and function of the cell. Thus DNA is the blueprint for life.
Ebright and his college roommate, James R. Wong, worked all that night drawing pictures and constructing plastic models of molecules to show how it could happen. Together they later wrote the paper that explained the theory. Surprising no one who knew him, Richard Ebright graduated from Harvard with highest honors, second in his class of 1,510. Ebright went on to become a graduate student researcher at Harvard Medical School. There he began doing experiments to test his theory.
If the theory proves correct, it will be a big step towards understanding the processes of life. It might also lead to new ideas for preventing some types of cancer and other diseases. All of this is possible because of Ebright's scientific curiosity. His high school research into the purpose of the spots on a monarch pupa eventually led him to his theory about cell life.
Richard Ebright has been interested in science since he first began collecting butterflies, but not so deeply that he hasn't time for other interests. Ebright also became a champion debater and public speaker and a good canoeist and all-around outdoors person. He is also an expert photographer, particularly of nature and scientific exhibits. How did you come with new ideas?
In high school, I was a straight A student. Because learning was easy, I turned a lot of my energy towards the debating and model United Nations clubs. I also found someone to admire, Richard A. Weeharer, my social studies teacher and advisor to both clubs. Mr. Weeharer was the perfect person for me then. I opened my mind to new ideas.
And that is one of the ingredients in the making of a scientist. Start with the first-rate mind, add curiosity, and mix in the will to win for the right reasons. Ebright has these qualities From the time the book The Travels of Monarch 10 opened the world of science to him, Richard E. Bright has never lost his scientific curiosity. By Robert W. Peterson.